on this computer. Okay, and then I'm going to go back and share the screen again, or at least I thought it was. Yeah, okay. Well, all right. Now, we all right, let's see. Annotate. Okay, we're on the last bullet of the plant growth and development concept here. Let me get this out of the way a little bit. And now, this actually involves, when you sense time or timing, it's most certainly includes a time of day. And I want to mention that sunflower is a good example of what we're talking about in terms of circadian rhythms or biological clocks. Heliotropism is what I'm describing here because right here at this moment, sunflowers in the morning, what happens is you have differential growth rates. In the morning, the stems elongate on different sides during the day, but in the morning they grow faster on the west side, the sun's on the east side, and it bends toward the light. And at the end of the day, the sunflower stem does the opposite, grows fat toward the west from, and so by the end of the day, they're facing back the way east again. And so the next morning, they're situated in the best particular spot. Now, it works better with young plants, but as the flowers mature and open, they settle into a stable east-facing orientation. So you pass a field of sunflowers, you can see them facing a certain direction. So that's within a given day. This is the concept slide we've seen before, but this time we're going to, let's see if I can make this, change this view and get rid of some of this stuff. Hide thumbnail view. I don't even need to look at myself. But now that we have looked, last time that we looked at plant hormones, which are phytohormones, literally plant hormones, and we looked at the movements or responses, like movements growth responses that plants have to these hormones. We now need to emphasize the dotted line part here. The responses would not happen without environmental influences. It goes back to anything is what it is for genetic reasons and environmental effects on the genetics. And that's now what we're going to look at, the environmental influences. And the dotted line reminds us of that in the concept map. We looked at the various kind of tropisms, phototropism, and gravitropism and thigmotropism, and we looked at nastic movements. And I don't know why I've got the question mark here, so don't ask me about that. I'm not sure why I've got it on this slide. And when you do this kind of thing in plant ecology, you're looking at abiotic factors. When you look at organisms competing with one another, that's biotic influence or biotic factors. But when you look at the environmental influences like that are abiotic, not living, abiotic, air, water, and et cetera, you're looking at abiotic factors, and it's a discourse called physiological ecology, also known as auticology, A-U-T-E-C-O-L-O-G-Y. Now, of all the sub-sciences in botany, ecology is not something that's broke out separately in this 300 and something slide set, but ecology is embedded in it. Bottom line is auticology or physiological ecology. When you look at one organism, you're dealing with auticology. My preference is to study the community. It's called synecology, community ecology, S-Y-N, another prefix that we'll talk about today when we talk about gametes. Syn means not S-I-N, S-Y-N means come together. And so scent ecology is community ecology, and alt ecology is when you look at the ecology of individuals, in particular, physiological ecology of an individual. Now for the deferred question, plants do what they do right on schedule in response to environmental cues like temperature and light. Photoperiodism down here has a lot to do with light duration in a 24-hour period. And we can have long day plants, short day plants, and plants that are neutral to the day length. And the flowering of the first two, long and short day plants, is determined by the effects of, actually, I guess the second and third, not the first two. The flowering is determined by the effects of red and far red light on two alternating forms of the phytochrome pigment. Phytochrome means, in Greek, plant color. Okay, and we'll talk about the fact in a moment that phytochrome is a protein, and it has two forms in terms of the wavelengths of light that it absorbs. But in fact, I'll go ahead and say right now that phytochrome is sensitive to red light in the 660 nanometer range of the visible light spectrum, and it's sensitive. Another form, it's sensitive to far red light in the 730 nanometer visible spectrum. Humans can barely see far red light, but they can see it. And many flowering plants use phytochrome to regulate the time of flowering based on the length of day. And actually, it's more about night, photoperiodism. We'll talk about it. Even though they call them long and short day, it's really the night. We'll point out in a minute that's so critical. And we mentioned earlier, and I'll mention again, fertilization is cold treatment, and some plants require it to flower. Like I mentioned, lilac is, you don't see lilac in the South because it just wouldn't flower because it's not cold enough. And as we'll find out in a minute, some of these plants, long and short day, require cold treatment on top of a certain light period. Or actually, the truth is, the dark period is, despite the name, it's really the dark period. Now, if we look at the relationship between three of the elements in the next slide, the timing for growth and development is an interplay between these three factors. Actually, I guess grammatically it should be among, between and among in English. I don't think they teach English anymore because nobody seems to. I've slipped up, and I should have known better. When it's more than two, you say among. So the interplay among these three factors. Photoperiod is the time per day that our organism is exposed to light. And temperature and light both play roles in the ability of a plant to track changes in day length and changing seasons too. And we've already said this, but I'll say it again. Many season responses of plants rely on the activity of the phytochrome photoreceptor, and it's a protein. Now, as, let's see. Yeah, now, in this slide, we're going to find out, besides the photoperiodism that we're talking about, plants, also, phytochrome does a heck of a lot of other things, quite a few other things. Many flowering plants use phytochrome to regulate the time of flowering based on the length of day and night, and that's photoperiodism, and to set circadian rhythms. And phytochrome is located in the leaves of most plants. And when you start talking about circadian rhythms, it's really proof or evidence of the biological clock that organisms tend to have. Phytochrome is found in most higher plants, and a form of it, a similar compound, has been found in a bacterium. Now, the reason I've got this little innocence plant here is indication that this plant normally blooms in February or March, but this thing was blooming way back in December. And you could argue that global warming might have something to do with it. I just went into CERNAC, SERNEC, and did a quick look at this plant and all the plants that ever been vouchered collected in that 233 herbarium database, and hardly any of them in the older days years ago bloomed as early as what's going on here. And so, but that has a lot to do. Well, again, the environment is changing, and that has a lot changing how the genes respond to it. Now, as we said, we just concentrated on photoperiodism. For years, everybody knew that plants respond to low levels of light by such things or a phenomena as we call eteliolation. ETIOLATION. And that is a fancy word. It's actually what's called New Latin for white. And what I'm talking about is in the cupboard, if you've ever had onions or potatoes and they're sprouting and they get the white appearance because of the reduced light. And that's what eteliolation is. And people just generally know about that, because it happens to everybody. It reminds me of a case one time. I had a cactus, a prickly pear, mounted on a herbarium sheet, and I had it in a herbarium cabinet. And it must have been a light crack in that cabinet because when I opened that thing up a year or so later, the daggum cactus sprang out and like to bit me. So, but or at least I felt it was. But it had actually grown in the dark and didn't die. It's hard to kill a cactus. I've got one in my front yard that I've accidentally hit with a lawnmower, and that sucker's still going. The fact that these responses are dependent on certain wavelengths of light is a more recent discovery based on studies of seed germination. And there's one of the factors that phytochrome gets involved in, seed germination. For example, it was observed that the seeds of some species won't even germinate in the dark despite being full of water. We call that imbibed, like I guess an alcoholic, imbibed with alcohol, full of water. Even though the seeds were full of water, they wouldn't germinate in the dark. Now, this has a practical application from the standpoint of the seed. And that means like seeds of some weeds in the fields would remain buried and dormant until they got exposed because somebody tilled the soil and turned the soil and disturbed the soil, and they got exposed to light. And so the weeds capitalize on that phenomenon. All right, red light is available to plants only during the day when the sun is shining. And far red light, though, on the other hand, is available both day and night. And that's where this slide comes in. And this, to me, is the most interesting thing that we're going to talk about on plant physiology today. Forget day-neutral plants and think about long day and short day plants. Let me take a sip of coffee here. Botanists still don't understand all the mechanisms that induce flowering, and I talked about florigen, and that's on page 176 of the Botany for Dummies. But phytochrome is involved in some of the long day, short day plants, and we know more about the phytochrome than we do about the florigen business. I said phytochrome is a protein. That's true. It exists in two forms. There's a 660 form of it for nanometers, and there's a 730 far red form of it. And when P660 converts to P730 when you expose it to red light. And on the other hand, the far red form, P730, changes to the 660 short form of red light when exposed to far red light. So and here's the important part, in darkness, the far red form of phytochrome 730 reverts to phytochrome 660. And so, and I said this, I alluded to this earlier, that though they're called long and short day plants, the critical period for this phenomena is in the darkness of the night. Okay, let's look at these two graphs. One is for a long day, one is for a short day. Notice this one goes to zero in terms of phytochrome 730 amounts, and this one does not. For a long day, which is what this one is, for a long day plant to flower, the night has to be short enough, and that's this shaded portion here, so that all of the 730, phytochrome 730, does not get converted. Okay, it doesn't get converted to 660. On the other hand, what's that?
Question, is the x-axis on those graphs, is that time of day, the 4
No,
o'clock, 8 o'clock?
the x-axis is a 24-hour day, yeah. Okay, yeah, in 24-hour period, yeah, 4 hours, 8 hours, 12. See, did that answer your question?
Yes, thank you.
Yeah, that's the time, it's the hours in the day. And obviously the shaded portion is the night portion. Now, all right, and even though this axis isn't labeled, this in the middle corresponds to the y-axis for both the phytochrome levels. Now, I'll repeat that, for a short day plant to flower, well, I didn't. I said for a long day plant to flower, the night has to be short enough, as you've seen here. This is a long night over. Whoops, what happened? This is a long night over here in terms of hours, but in this night being short, then and that enables some of the phytochrome 730 to still be there. So there needs to be some to always be present for a long day plant to flower. On the other hand, a short day plant, the night is long enough to get rid of all of the 730 and convert it to 660 for it to flower. Now, we'll see black henbane in a couple of slides over as an example of a long day plant. Winter barley is another example. Beet, Beta vulgaris, is a long day plant. A short day plant, there are two examples I can think of. One of them is a ragweed, Ambrosia artemisifolia, and cocklebur, Xanthium sumerium. Corn is a day neutral plant. Cucumber is a day neutral plant. Phytochrome has no effect on those plants in terms of flowering. Now, we won't go over all this because we don't have enough time in detail. We won't have enough time to go over all of it. The first one has to do with something. Well, here, let me go ahead and show you this. This is a long day plant that's called henbane. And I said that some plants have to have cold treatment, and this is one of them. It has to be subjected to cold flower in addition to have that long day or actually short night regimen. And to make matters more complicated, this is genetically controlled in the sense that henbane produces an annual and a biennial type of plant. The annual strain produces flowers in the first growing season, and the biennial type requires a cold winter before the flowering takes place in the second growing season. So that experiment has a lot of variables. Now, back to this. I already said the Florigen business is still some unknowns there. We don't know what it is. Theoretically, there is a compound called Florigen. And the ability of a plant to flower is obviously affected by the maturity of the plant. We've talked about biennials taking 2 years to flower. In fact, besides mentioning it a moment ago, we mentioned annuals, perennials, and biennials some time ago. And though it doesn't flower, ginkgo is a conifer or related to conifers. It doesn't have a well-defined cone for the female. The male is kind of in a cone-like arrangement, but it doesn't produce flowers. It does produce reproductive structures. And ginkgo takes 20 to 40 years to reach sexual age or puberty, at least for the female. The plant can live supposedly for 1,000 years. Ginkgo was found as a fossil before it was found alive in China. And so what we have in this country today that's planted came from there. Now, Donna has a female in ginkgo in her yard that was there, I think she said, when she bought the property. But and she knows, and anybody else knows that's got one, the female has an integument around it that looks like a fruit, but it's really a fleshy integument. When I say integument, think about the red skin on a peanut, Spanish peanut. That's an integument. It was actually at maturity, it's a seed coat. But the fleshy integument on a ginkgo turns rancid, and you can smell it for a city block. So the female, unfortunately, is not real desirable. And I haven't asked Donna, but I bet it stains stuff also. I went out in the yard to get the cycad, which has a fleshy integument too, but the seeds aren't. The cone hadn't opened up yet enough for me to get one out. I'm not going to bust it open just for this class, but it's a fleshy orange integument on a lot of cycads, really fleshy, kind of like ginkgo. Now, some desert annuals like Arabidopsis thaliana is a mustard family, Arabidopsithaliana, completes the life cycle in 40 days. And that's a lot less than 1,000 years. And the shortest plant on record that I know of that goes from seed to seed, and I don't remember the exact plant right now without looking it up, but there is a plant in the Saharan desert that goes from seed to seed in 10 days. And what it is just capitalizing on what little bit of moisture there is when there is moisture, and doing its thing when the time is right. Now, I've got these two here, and I'm going to talk about these, and then we're going to move on into getting close to reproduction, and this is useful for reproduction. Fertilization is a synonym for oogamy, a special kind of gametic union. But I want to distinguish between the two. First of all, you're not going to get fertilization unless you have pollination. And I could ask you what pollination is, but in angiosperms, it's a little different than gymnosperms. It's easier to understand for flowering plants and conifers. Pollination is a transfer of pollen from the stigma of the male of the stamen transferring a pollen from the anther, which is the mycosporangium of a flowering plant, to the stigma of the female part. That's what pollination is, transferring pollen from the anther to the stigma. And it's more complicated in the gymnosperms to describe it, but it's transferring pollen from the pollen cone, actually from the little tassel that gets all the pollen on your car, transferring the pollen from there. And actually, I'm going to go ahead and show that now. Oops, this one here. Nope, not. Okay. A while back, I showed this. Steve and Ronna asked me to send them this and send me the counterpart, the male. These were slides that I threw in. This is a longitudinal section, and I'm still explaining the difference in pollination fertilization. I'm talking mostly about pollination now. In a gymnosperm, it has to do with transferring pollen from this. Here's a little. This is a pollen cone. It's about an inch long, maybe. A section through the middle of it, mounted on a slide. And there's a sporophyll. Donna brought up the term sporophyll from the matching, which is a structure that supports spores. And in this particular case, the spore case, the microsporangium is on the underside. Whereas those are pollen grains in there. have little Mickey Mouse-like looks to them, ears that fly around, help it to fly around. And that pollen would fall down in the cracks of this female cone, which is about an inch long. Everybody thinks of cones. When you see a cone, you're looking at something that's 2 years old. When that sucker is pollinated, it's only an inch high maybe, and this is about an inch, mounted on a glass slide in a midsection. So everything is actually, it's pretty good to get as many of these attached as you could in a midsection, because you're slicing a three-dimensional structure. So some of them are attached and some of them are not attached. You have to think 3D. And that's where microscope work and lab comes in handy. And that's the stumbling block for a lot of people. They just can't get using the microscope and think 3D. Embryology of animals helped me to get this concept down, think 3D. But all right, so here's in year one, the pollen lands down in here, and we're going to see a bigger version of this in a little bit when we start talking about reproduction. But it takes a year for that pollen tube to grow in through this megasporangial wall. No, the female. Okay, so let's go back. That's pollination. Okay, that's pollination. Now, fertilization is simple. It's where running bear meets little white dove, so to speak. And Steve knows that joke. There's a song associated with that. But basically, it's where two gametes meet. And in the process, you think of it as fertilization. You got to have pollination before you have fertilization. Now, I'll repeat it probably again, but in some gymnosperms like pine, the period between pollination and fertilization is a year, so the whole cycle takes 2 years. Juniper is also a conifer. It takes a year to do its cycle. All right, before we leave this god-awful topic of plant physiology, let's talk about the fact that temperature, an abiotic factor, not only affects flowering, but affects vegetative growth also. Back in the 70s, I was at Penn State, and there was this underground heating duct system, and the part that was associated, you could tell where the heating duct is. In the middle of winter, you get the grass being green and healthy, and over here it looks typical of what it looks like it when it's not heated. And I'm going to define cardinal temperatures on this slide. There's three phenomena here. There's a minimum temperature, a maximum temperature, and an optimum range. And different plant organs can have different cardinal temperatures. Roots generally have lower cardinal temperatures than stems or shoots. And we can talk about the range of temperatures for organisms, and this would apply to bacteria too, as three cardinal or pivotal temperatures or regimes, because two of them are temperatures, this one and this one, and this one is a range or regime, optimum. The maximum, well, let's do the minimum first. The minimum temperature is the lowest temperature that allows continued growth and metabolism. That's the minimum. The maximum temperature is the highest temperature at which growth and metabolism can still proceed. And if you get above the maximum, growth will stop, and eventually it continues to rise beyond that point. You start, the proteins and enzymes and nucleic acids in the organism get damaged, denatured, and destroyed, and death results. I call that the 4Ds, okay? Damage, denaturization, destruction, and death will result. Now, the optimum range is the small temperature range between the minimum and the maximum that promotes the fastest growth rate, which means another way of saying the shortest generation time for a metabolism in a given species. All right. Okay, any questions about plant physiology?